In 2017, a team of researchers published the first successful ancient DNA study of Egyptian mummies, and the findings overturned assumptions that had been built into Egyptology for over a century. The study was published in the journal Nature Communications. The researchers, led by scientists from the University of Tübingen and the Max Planck Institute for the Science of Human History, analyzed genetic material from 151 mummified individuals recovered from the archaeological site of Abu Sir el-Melek in Middle Egypt. These individuals had lived and died across a span of approximately 1,300 years, from around 1380 BC to 425 AD, a period covering the New Kingdom, the Ptolemaic Greek period, and the early Roman era. The technical challenge was formidable. Egypt's hot, humid climate is one of the most hostile environments for DNA preservation on Earth. Organic material degrades rapidly at high temperatures, and the chemical processes used in ancient Egyptian mummification, including salt treatment and resinous coatings, further fragment and contaminate genetic material. Previous attempts to extract ancient DNA from Egyptian mummies have been plagued by contamination issues and technical failures that left their results unreliable. The 2017 team used new extraction protocols targeting the petrous bone and dental pulp, the same dense inner ear bone and tooth root structures that have been most productive in ancient DNA studies elsewhere. Combined with rigorous contamination controls and statistical methods to distinguish genuine ancient sequences from modern laboratory contaminants. The results were unambiguous, and one finding in particular stopped the academic world in its tracks. Before 2017, attempts to extract ancient DNA from Egyptian mummies had produced results that most scientists rejected as contaminated by modern handling. The 2017 study was the first to meet the rigorous methodological standards of modern ancient genomics, and its publication in Nature Communications, one of the most prestigious scientific journals in the world, meant that its findings could not easily be dismissed. The ancient Egyptians of this period were genetically most similar not to modern Egyptians or to sub-Saharan Africans, but to ancient and modern populations from the Near East. Specifically from the Levant, the region encompassing modern Israel, Palestine. The 2017 study's legacy is not just in its findings, but in the methodology it established, making ancient Egyptian mummies a legitimate subject of ancient genomics for the first time. The DNA evidence from the 2017 study places ancient Egyptian origins firmly within the context of the broader ancient Near Eastern world. But to understand what this means, it helps to trace the genetic story back further, to the migrations and population movements that established the Nile Valley as a center of human civilization in the first place. The Nile Valley was not populated in isolation. The river corridor connecting sub-Saharan Africa to the Mediterranean coast was one of the major corridors of ancient human movement in the Old World, used by populations moving both northward and southward across thousands of years. But the DNA evidence suggests that the dominant ancestral contribution to the ancient Egyptian population came from the north and east, from the expansion of Neolithic farming communities out of the Fertile Crescent and the Levant that began around 10,000 years ago. The difference between Upper Egypt, the southern upstream portion of the Nile Valley, and Lower Egypt, the northern delta region, is genetically detectable, even in ancient DNA. Upper Egyptian populations show somewhat higher levels of sub-Saharan African ancestry, consistent with their more southerly geographic position and their proximity to the migration corridors from the African interior. Lower Egyptian populations, including those at the Abu Sir el-Melek site, show more pronounced Near Eastern genetic affinity. The genetic distinctiveness of pre-dynastic and early dynastic Egypt, the period when the great civilization was forming, is now being studied through new ancient DNA work at sites predating the Abu Sir el-Melek sample. Early results suggest that the founding genetic profile of Egyptian civilization was established by the mixing of indigenous northeastern African hunter-gatherer populations with incoming Near Eastern Neolithic farmers, producing a population that was biologically distinct from anything else in Africa or the Near East, while being closely related to both. The Nile itself was the enabling factor in all of this. As one of the world's great rivers, the Nile provided the water, the agricultural fertility, and the transportation corridor that made civilization possible in what is otherwise desert. The genetic diversity of the ancient Egyptian population reflects the Nile's role as a biological highway, drawing people from the north and east, from the south and west, and mixing them in the narrow strip of habitable land along its banks. The land of Punt, the distant trading partner that Egyptian texts reference as a source of incense, ebony, and exotic animals, has been located by most modern scholars in the region of modern Eritrea, Djibouti, or Somalia. The Egyptians did not just practice royal incest, they elevated it to theology. The justification was embedded in the mythology of the gods themselves. Osiris and Isis were siblings who married and produced Horus. The divine family at the center of Egyptian religious belief was an incestuous one, and the imitation of divine behavior was not merely permitted for the royal family, it was, in their cosmological framework, the defining obligation of divine kingship. The pharaoh was not a human king, he was a living god. Gods married their sisters, therefore so did pharaohs. The genetic evidence for how this played out in practice, and what it cost in biological terms, comes most dramatically from the DNA analysis of the 18th dynasty royal family, conducted in a 2010 study published in the Journal of the American Medical Association. The study analyzed DNA from 11 mummies, identified as belonging to the family of Tutankhamun, the boy pharaoh who died around 1323 BC at approximately 19 years of age. The results confirmed a family tree of striking genetic density. Tutankhamun's parents were identified as full siblings, both children of Amenhotep III and his great royal wife, Ti. The degree of inbreeding in his immediate ancestry was extreme, even by the standards of ancient royal incest. The physical consequences are documented in the same study. Tutankhamun had a severe club foot. One foot was so badly deformed that he could not walk without a cane, and over 130 walking sticks were found in his tomb. This seemingly minor archaeological detail, 130 canes, became one of the most powerful pieces of evidence confirming the DNA findings. Why would a young pharaoh need 130 walking aids unless he genuinely could not walk without assistance? The physical evidence and the genetic evidence told the same story. He had choler disease, a bone condition affecting the foot. He had a cleft palate, and he carried 2 separate strains of malaria. The combination of congenital defects and disease almost certainly contributed to his death at a young age. The genetic damage accumulated through generations of close relative marriage was, in the most powerful royal family in the ancient world, leaving consequences that the DNA could still read 3,000 years later. The Akhenaten question remains one of the most debated in Egyptology. Few questions in the history of Egyptology have generated more political heat and less scientific clarity than the question of the skin color of ancient Egyptians, and specifically of the pharaohs. The debate has, for most of its modern history, been entangled with contemporary racial politics in ways that have complicated the ability to have a purely scientific conversation. Afrocentric scholars have argued, with significant passion and some historical evidence, that ancient Egypt was a black African civilization whose cultural and intellectual achievements have been systematically appropriated by European scholars who depicted the ancient Egyptians as Mediterranean or Middle Eastern in appearance. European and American mainstream Egyptology has historically defaulted to a Near Eastern or Mediterranean characterization, without always acknowledging the African geographic and cultural context of Egyptian civilization. The ancient DNA evidence provides important but not conclusive data on this question. The 2017 Abu Sir el-Malek study confirmed that the ancient Egyptians of the New Kingdom and later periods carried genetic profiles consistent with Near Eastern and Northeastern African ancestry. The study's lead authors were careful to note that their sample, from a single site in Middle Egypt covering a specific time period, could not be used to characterize all ancient Egyptians from all periods and all regions. Egypt is a long, geographically diverse country, and the genetic composition of its ancient population varied across both space and time in ways that a single study cannot fully capture. With limited sub-Saharan African contribution, but sub-Saharan African ancestry was not absent. It was present, simply at lower frequencies than in modern Egyptians. And the study covered only one site in Middle Egypt over a specific time period. Generalizations about all ancient Egyptians from all periods are not scientifically warranted. Ramses II, the most celebrated pharaoh in popular culture, the likely subject of the Exodus narrative in various traditions, provides one of the more specific data points in this discussion. Analysis of his mummified hair, conducted in 1976 by French scientists, found that the natural hair color of Ramses II was red. Not dark brown or black, red. The red pigmentation in his hair was identified as pheomelanin, the same compound responsible for red hair in living people. Whether this indicates genuine genetic red hair coloring or whether the mummy's hair had oxidized over time to produce a reddish appearance is still debated. But the finding contributed to a more complex and less stereotyped picture of what the royal family of ancient Egypt actually looked like. The mummification process is one of the most technically sophisticated procedures developed in the ancient world, and the byproduct of its elaborate chemistry has, paradoxically, made it both easier and harder to recover DNA from the resulting remains. The standard mummification process, as described in Greek and Roman accounts and confirmed by physical analysis of mummies, took approximately 70 days. The first step was removal of the internal organs through an incision in the left side of the abdomen. The liver, lungs, stomach, and intestines were removed separately and placed in canopic jars for preservation alongside the body. The brain was removed through the nasal passage using a long hook, a procedure that sounds brutal but left the skull intact for preservation. The heart was left in place, as Egyptian theology held it to be the seat of consciousness and the organ that would be weighed against a feather of truth in the afterlife judgment. The body cavity was then packed with natron, a naturally occurring sodium salt found in Egyptian desert deposits, and the body was buried in natron crystals for approximately 40 days to thoroughly desiccate the tissues. After drying, the body was cleaned, anointed with resins and aromatic oils, and wrapped in hundreds of meters of linen bandages in a specific pattern. The chemical analysis of the resins used in mummification has produced remarkable evidence about Egyptian trade networks. Isotopic and molecular analysis of resin residues from mummy wrappings has identified substances derived from cedar trees, which grew in Lebanon, not Egypt, from pistacia trees, from the Middle East, and from plants whose origin points to sub-Saharan Africa and possibly even South Asia. The pharmacopoeia of mummification was international, sourced from trading partners across thousands of kilometers of ancient commercial network. The linen used in mummy wrappings was consistently of very high quality, a type of fine linen that required specialized agricultural and weaving knowledge to produce. Chemical analysis of the linen fibers has identified specific varieties of flax cultivated in Egypt's Nile Delta, confirming that the Egyptian linen industry was a major economic activity whose supply chains were deeply integrated with the agricultural and craft production systems of the Nile Valley. The claim that cocaine and nicotine were found in Egyptian mummies both plants native to the Americas, has generated intense debate since German researcher Svetlana Bala. The story of ancient Egypt is, in the conventional telling, a story of pharaohs, kings, male rulers, the names carved on temple walls and inscribed in cartouches. The real story is considerably more complicated, and the evidence from both DNA analysis and archaeology has helped restore the prominence of the women who shaped Egyptian history. Hatshepsut is the most dramatic example. She ruled Egypt as a pharaoh, not as a regent or queen consort, but as a full pharaoh with the male titulary, depicted in official art wearing the double crown and the traditional false beard. For approximately 2 decades, from around 1479 to 1458 BC. She was one of the most prolific builders in Egyptian history, responsible for the mortuary temple at Deir el-Bahri that remains one of the architectural masterpieces of the ancient world, and for the expedition to Punt, documented in its famous wall reliefs. After her death, a systematic campaign erased her from the official record. Her statues were demolished, her name was chiseled from inscriptions. Her image was replaced by that of her successor and stepson, Thutmose III. For over 3,000 years, she was effectively invisible. Her rediscovery in the 20th century was gradual. The physical identification of her remains, confirmed through an extraordinary piece of forensic work in 2007, came from a single molar tooth found in a wooden box inscribed with her name. The tooth's roots fit precisely into the jaw socket of an unidentified female mummy that had been in the Cairo Museum collection for decades. The mummy was identified as Hatshepsut by this dental match, not by DNA analysis. Her genetic material has not been successfully recovered, but the identification has been widely accepted. Nefertiti, the wife of the controversial pharaoh Akhenaten, remains one of the most recognizable faces from the ancient world, primarily through the famous painted limestone bust in the Berlin Museum. Her tomb has never been found. Akhenaten himself is one of the most genetically debated figures in Egyptian royal history. His elongated skull and unusual body proportions, depicted consistently in the art of his reign, have been proposed as evidence of genetic disorder caused by royal inbreeding, possibly Marfan syndrome or Antley-Bixler syndrome. The DNA evidence from his mummified remains, analyzed as part of the 2010 JAMA study, identified him as Tutankhamun's father, but did not definitively resolve the question of his physical appearance. The ancient Egyptians were, by the standards of their era, sophisticated medical practitioners. The Edwin Smith papyrus, dating to around 1600 BC, but likely copying earlier texts, contains clinical descriptions of injuries and diseases that are recognizable to modern physicians, presented without reference to supernatural causes. This document-based rationalism in Egyptian medicine coexisted with a vigorous tradition of magical healing, but its existence demonstrates that Egyptian physicians were capable of empirical observation and systematic thinking about the human body. The mummies have turned out to be one of the most valuable archives of ancient disease in the world. CT scanning of mummy collections has revealed atherosclerosis, hardening of the arteries, in a significant proportion of ancient Egyptian mummies, including individuals who lived active, physically demanding lives. This finding, published in a 2011 Lancet study, challenged the assumption that cardiovascular disease was a modern lifestyle condition, and suggested that it has deeper evolutionary and dietary roots than previously recognized. Malaria. DNA has been recovered from Egyptian mummies dating as far back as 3,500 years. Among the earliest confirmed cases of the parasite Plasmodium falciparum in the ancient world. The presence of malaria in ancient Egypt is consistent with the ecological conditions of the Nile Delta, where standing water and warm temperatures created ideal breeding conditions for the Anopheles mosquito. The use of moldy bread as a wound treatment, documented in several ancient Egyptian medical papyri, anticipates the antibiotic principle by over 3,000 years. The penicillium mold that grows on bread produces penicillin, and while the ancient Egyptians could not have known this, their empirical observation that moldy bread reduced infection and promoted wound healing was clinically correct. The curse of the pharaoh. The popular belief that those who opened Tutankhamun's tomb in 1922 were struck by a mysterious ancient curse has been subjected to scientific analysis. Studies of the mortality records of the excavation team found no statistically significant elevation in death rates compared to age-matched comparison groups. The curse is not supported by the evidence. The traditional Egyptian medical procedure of trephination, drilling or cutting holes in the skull, is documented in several mummified individuals. Unlike in many other ancient cultures, where trepanned individuals rarely survived the procedure, some Egyptian cases show healed bone around the opening, indicating that these patients lived for months or years afterward. The skill of Egyptian surgeons was, by ancient standards, genuinely remarkable. Ancient Egypt was not a closed system. For 3,000 years of pharaonic civilization, the Nile Valley was repeatedly entered by outside forces, military, commercial, and demographic, and each major contact left a trace in the population's genetic profile. The Hyksos, the name means rulers of foreign lands in Egyptian, arrived in Egypt from the Levant sometime around 1650 BC and established control over Lower Egypt for approximately a century, founding the 15th dynasty. For centuries, Egyptian sources depicted the Hyksos as violent invaders who destroyed temples and disrupted civilization. Ancient DNA analysis of skeletal remains from the Hyksos capital at Tel el-Daba in the Nile Delta, published in 2020, found that the Hyksos rulers showed genetic profiles consistent with Levantine origin, confirming their Near Eastern heritage, but also revealing that the takeover was not a sudden military invasion. The genetic evidence suggests a gradual process of immigration, social integration, and eventual political takeover, rather than a single violent conquest. The Hyksos brought the horse-drawn chariot to Egypt, a military technology that Egypt subsequently adopted and deployed to build its own imperial power in the New Kingdom. The DNA evidence from Tel el-Daba showed something else remarkable. Even before the Hyksos period of political control, individuals with Levantine genetic ancestry were present in the lower Nile Delta, suggesting that immigration from the Levant into Egypt was a long-term demographic process rather than a sudden military event. The political invasion was the culmination of a gradual population movement that had been occurring for generations. They also introduced the composite bow, vertical loom weaving, bronze working techniques, and musical instruments including the lyre. The conquerors, once conquered by Egypt in the 16th century BC, left a lasting material and technological legacy. Alexander the Great's conquest of Egypt in 332 BC introduced the Macedonian Greek ruling class, the Ptolemaic dynasty that culminated in Cleopatra VII, whose genetic profile was entirely European. The Ptolemies deliberately adopted Egyptian religious and royal traditions while maintaining their Greek cultural identity. Cleopatra was the first Ptolemaic ruler to learn the Egyptian language. The DNA of the Greek ruling class affected the upper strata of Egyptian society, but had limited impact on the broader population's genetic profile. The Roman period that followed the Ptolemaic dynasty introduced yet another genetic layer: Roman administrators, soldiers, and merchants who settled in Egyptian cities. The everyday life of ancient Egypt was, by the standards of the ancient world, unusually well documented, through papyri, through tomb art, and now through the biological evidence that DNA, isotope analysis, and physical examination of mummies have added to the record. And the picture that emerges is both more familiar and more alien than the monumental architecture of Karnak and Luxor suggests. The ancient Egyptians' relationship with animals was unlike anything in the ancient Western world. Cats were associated with the goddess Bastet, and killing one, even accidentally, was a capital offense in some periods. The cult of the sacred cat extended to mummification. Millions of cat mummies have been recovered from Egyptian temple sites, along with mummified crocodiles, ibises, baboons, and bulls. A 2019 genetic study of ancient Egyptian cat mummies, conducted by the Royal Belgian Institute of Natural Sciences, traced the origin of the domestic cat, now found in homes on every continent, to a lineage that spread from ancient Egypt to the rest of the world during the pharaonic period. The black eye makeup that both men and women wore in ancient Egypt, the distinctive kohl that defines the visual signature of Egyptian portraiture, was not purely cosmetic. Chemical analysis has shown that the lead-based compounds in ancient kohl have antibacterial properties and may have reduced the rate of eye infections in the dusty, fly-rich environment of the Nile Valley. The practice was functional as well as aesthetic. Female tattoos in ancient Egypt, documented in mummies dating to the Middle Kingdom and now confirmed in the DNA-analyzed individuals of the New Kingdom period, appear to have been associated with fertility and protection during childbirth. The patterns, dots and dashes on the lower abdomen and thighs, are concentrated in areas that would have been relevant to pregnancy and labor. The tattoos were not decorative in the modern sense. They were protective technology. The oracle system, in which questions were posed to divine statues whose priests manipulated them to produce yes/no answers, was a functioning judicial mechanism in some periods. Legal disputes could be submitted to the oracle of a god, and the oracle's movement toward or away from a petitioner was recorded as a binding decision. The oracle of Amenhotep I at Deir el-Medina, the workers' village whose DNA archive has been extensively studied, was consulted on theft accusations, labor disputes, and questions of property inheritance. Deir el-Medina itself is one of the most DNA-rich archaeological sites in Egypt. Let's bring the full picture together, because the ancient DNA story of Egypt is not just about one civilization. It is about the most studied civilization in the history of archaeology, finally being understood at the level of the individual human beings who built it. The genetic legacy of ancient Egypt in the modern world is concentrated most clearly in one specific population, the Coptic Christians of Egypt. The Copts are the direct descendants of the pre-Islamic Egyptian population, the people who maintained the Egyptian Christian tradition through the Arab conquest of 641 AD and the subsequent Islamization of the Nile Valley. Genetic studies of Coptic communities have confirmed that they carry elevated levels of the ancient Egyptian genetic profile, specifically the Near Eastern and Northeastern African ancestry that the 2017 mummy study identified in its ancient samples, compared to the broader modern Egyptian Muslim population, which shows higher levels of Arabian Peninsula and sub-Saharan African ancestry, acquired through the Islamic period. The Copts are, in a measurable biological sense, the closest living relatives of the people who built the pyramids. The 2017 Nature Communications study demonstrated one additional finding of particular significance. The sub-Saharan African component of modern Egyptian DNA is substantially higher than it was in the ancient mummies. The researchers estimated that the increase in sub-Saharan African ancestry in the modern Egyptian population represents approximately 8% of the total genome, accumulated primarily after the Ptolemaic period, likely through the trans-Saharan slave trade of the Islamic era, which brought large numbers of sub-Saharan Africans into North Africa over approximately 1,000 years. The question of race, of what color the ancient Egyptians were, what they looked like, how they should be represented in museums and films, is one that the DNA cannot fully answer, because skin color is a polygenic trait, whose expression is not captured completely by the ancestral population categories that genomic research uses. What the DNA does tell us is that the ancient Egyptians were a genetically distinct people, biologically connected to their Near Eastern neighbors, but shaped by the specific ecology, history, and cultural isolation of the Nile Valley into something unique. They built for eternity. The DNA confirms that their biological line, in modified and dispersed form, has achieved something close to it.